Hi, everyone. Welcome to Foresight's Neurotech Salon series. I'm really excited today to have Markus Kaiser here. And Markus, you'll be discussing improving cognition in mental health through non-invasive ultrasound neuromodulation. Thank you so much for joining us. Real pleasure to have you. And yeah, please take it away, and I'll share a lot more info about you in the chat.
Okay, thanks so much for the introduction and for the invitation to give a talk within this series. So you should all be able to see my screen. Yeah, I'm based at Nottingham University, the School of Medicine, and I'm also co-director of the Institute of Mental Health Center for Translational Mental Health. So really try to develop new technology, and in particular brain stimulation technology that can be used later on for clinical applications. Nottingham is known for two things. One is Robin Hood. I'm not talking about Robin Hood today. And the other one is inventing MRI. So the Nobel Prize for inventing MRI went to Sir Peter Mansfield, who invented this at Nottingham University. And because of that, we're also hosting the National MRI Center in the UK. So we are getting an 11.7 Tesla human MRI scanner next year, which means we will have higher spatial resolution to see what is changing in terms of brain structure and function in healthy participants and in patients. And we are launching a center for new technology, new modulation, and new therapeutics early next year with 24 faculty members. So it's not only neuroimaging with MRI or EEG, but it's also mobile MEG, which is called OPM. So there's a Nottingham startup company, Saka Magnetics, that has a mobile MEG device. So it can be used in children. They can move the head around, and you can still measure MEG in participants. And we're one of the few centers in the UK that use focused ultrasound. And the idea is to use information from imaging about how the brain is connected to inform what is actually happening after ultrasound stimulation and where should we stimulate. So what you see here is connectivity in a human participant. The colors tell you the local direction of a fiber tract. Green means going from the back to the front. Red means going from left to right or right to left. And blue means going from the top to the bottom, from the bottom to the top. So unfortunately, we don't know about directionality based on diffusion tensor imaging, based on MRI data. But just based on connectivity, we can predict the age of a participant plus minus 8 years, or intelligence plus minus 3 to 5 IQ points. So we can already have some idea of what is going on in the system, and we use this as a starting point to predict what happens after brain stimulation. So how are changes spreading through the network, and what cognitive changes would we expect based on brain stimulation in individual participants? So the idea is to use connectome-informed, personalized neuromodulation for brain and mental health. So what's stopping us to use brain stimulation in human participants? And there are several questions that need to be answered before you perform brain stimulation. One is, which part of the brain should be targeted? So if you parcellate the brain, let's say, into 100 regions, which of those regions should be stimulated? Or if you stimulate fiber tracts, which of the 1,000 or so fiber tracts should be stimulated in the brain? And then how to stimulate? So what is the optimal stimulation protocol for an individual participant? And ideally, you would also like to know what are long-term effects. So what happens hours or days or weeks after stimulation? And in particular, what could be negative side effects over the long term? So you don't want to have an intervention that treats anxiety and then half a year later, someone starts to have epileptic seizures. So you don't want to have negative long-term effects as a consequence of brain stimulation. Ideally, you would like to have computational models that can predict immediate and long-term effects based on what is happening. And you would also like to have non-invasive approaches. Why non-invasive approaches? We did a survey with almost 800 participants, half of them healthy participants and half of them had a mental or brain health condition. And we were asking them, what kind of intervention would you prefer, either because you have a condition at the moment or if you would have a brain condition in the future. For the patients, many of them had Parkinson's disease, but we also had other participants with depression, psychosis, migraine, different kinds of disorders in the mix here. So we gave them 5 different interventions, and we were asking, which ones would you prefer, which ones do you think are safe, and which ones do you think are very efficient in terms of changing brain dynamics. And this is the pattern we see over here on the right-hand side. If we ask them which is the preferred intervention, people opted for ultrasound. So possibly they thought if it's safe for pregnancy scans, it's safe for brain stimulation. And the second place was magnetic non-invasive brain stimulation with transcranial magnetic stimulation. The third place was pharmaceutical drugs. So people know pharmaceutical drugs, but they also know that there were side effects for pharmaceutical drugs. Then you have electric stimulation. So for electric stimulation, people might just think about One Flew Over the Cuckoo's Nest and electroshock therapy. So people didn't like electric stimulation. But what people really didn't like was brain implants. So you can see on the left-hand side, you have this figure here. So many participants were saying that they will never use brain implants. So both for patients and for healthy participants, there was a sizable chunk of people who said they would never use it. If we then look at why that is the case, ultrasound and magnetic stimulation is perceived as safer than other options. If we look at effectivity, ultrasound is seen as effective, but also implants are seen as very effective. So the reason that implants are not the preferred option is because of the safety. It's not based on they're less efficient than other modalities. Okay. So based on safety, the preference is really for non-invasive technologies, in particular for ultrasound and magnetic stimulation. So what is ultrasound? You basically have this device on the left. It just looks like an oscilloscope on a bench. So it's a very small device. And you can basically use this device to stimulate the brain. You can go several centimeters deep. This device has been on the market since October 2019. And in the middle, you can see that you can go directly for deep brain structures. You can see changes in mechanical pressure at the target. And this pressure is really localized. So you're not stimulating parts that are in your pathway, but you're only stimulating at the focal area. There are several centers in the UK at the moment, Plymouth, Nottingham, Oxford, Cambridge, and there are more centers coming up like Yale, Brighton, Glasgow, Bristol, and so on, currently getting that equipment. So the benefits are that ultrasound is very focal. You can directly target deep brain structures and is non-invasive. And depending on the protocol, you can either increase or decrease brain activity. And it's also possible to measure MRI or EEG responses while you're stimulating. So the sound wave is not interfering with MRI and it's not interfering with EEG. So you can basically measure the reaction of the brain at the same time while you're stimulating. What you see here is an ancient slide from a long time ago, so up to 2020. Most of the studies came up in the last couple of years because it's such a novel technology. But if we just go back to 2020, you can see some examples of what has been tried. There have been studies using ultrasound stimulation along with transcranial magnetic stimulation. There have been tests to change mood in healthy participants and also changing mood in mild to moderate depression patients. There have been studies on traumatic brain injury, on changing tactile sensations and temporal lobe epilepsy, and so on. And in the meantime, there have been a lot more studies looking at anxiety, looking at schizophrenia, looking at many different conditions. And just to give you an idea what might be possible in the future, you might know about the InsideTech system. The InsideTech exoplate system is used for brain surgery. So it can basically send ultrasound pressure into a focal area. Rather than having one sound source, you have 1024 sound sources, and they're all converging onto a very small part of the brain. So it's a resolution of around a cubic millimeter or even below that. And because you put so much pressure into the focal area, you're basically heating up the tissue and you're destroying brain tissue. However, in this study by Jean-François Aubry, he was using a special protocol for the InsideTech system, so that only he is using at the moment in Paris. So rather than turning the system to 11, he was basically only very low amplitude of ultrasound. And he was targeting the VIM target here. And what he saw was a 98% reduction in tremor for essential tremor patients for at least 20 minutes. Why not longer than 20 minutes? Because the surgeons wanted to fry up the tissue. So at least before the surgeon started, he was able to record for 25 minutes, and there was this almost complete extinction of tremor in patients. So in the future, it might be possible to have systems that can reduce tremor without having a brain implant for patients later on. So that was published earlier this year. So what is ultrasound actually doing? So you can see an ultrasound transducer applied to the head. So you need some coupling, some ultrasound gel between the skin and the transducer. And you then target some deep brain structure in the focus. And the idea is that there were some channels in the cell membrane that are reacting to mechanical pressure. So when the ultrasound arrives, you have those channels opening, you have changes in internal calcium, and then you have this second messenger cascade where other ion channels show changes. And because of that, you see changes across the membrane, which makes it more likely or less likely for a neuron to fire. So depending on your protocol, you can either have an inhibitory effect or an excitatory effect. So the idea is to have connectome-informed, personalized neuromodulation for brain and mental health. And we started to test some of those ideas in rhesus monkeys. So what you see here is an experiment done in Oxford by Jérôme Salé. So we have 3 monkeys, they're anesthetized, and we have 3 different targets: anterior medial prefrontal cortex, supplementary motor area, and posterior anterior cingulate cortex. And we basically observe what is changing in terms of fMRI activity after stimulation. So we do this for all 3 targets, but if we look at one of those targets, the top target here, we can observe changes in fMRI and in functional connectivity in other parts of the network. So we're not observing at the target region, but we observe at other connected regions within the brain. So what you see here is time courses and functional connectivity in 3 other regions. And for the first region, you see this early peak, and then functional connectivity change goes down again. For another region in the middle, you see a later change, so maybe 10 minutes later or so. And for the third region at the bottom, you see an early change, but then 20 minutes later, you see an even higher peak change in functional connectivity. So this means that if you have a snapshot at 3 different time points, you get different results. So you might have one lab that is only looking at the first snapshot. So they would say the first area in green is activated. So you see changes after ultrasound. And another lab is measuring 10 or 20 minutes later, and they don't report a change. So they say there's no effect in that area. So this means it's really important to observe fMRI over a longer time frame to be sure that you're really observing what is happening induced by focused ultrasound. And we then moved on to human studies. So this is a study done here in Nottingham, where we have healthy human participants. We have a baseline session where we have resting state fMRI, and we look at resting state functional connectivity. We then have participants performing a stop signal task. So you're presenting a green circle, and then people are supposed to press a button when they see the green circle. But while they're about to press the button, we either leave the green circle on or we change to red cross. And if the red cross is appearing, they have to stop pressing the button. So they have to inhibit themselves not to press the button. So it's a cognitively demanding task. And we measure the reaction time, how fast are they in pressing the button, but we also measure the accuracy, whether they make any mistakes. Then we have the structural MR data to plan our ultrasound experiment. So we predict where we should put the sound source, the transducer, in order to stimulate the target. And we then have a second session where we have focused ultrasound stimulation. We then again look at resting state functional connectivity, and we then perform the stop signal task. And we perform the stop signal task around 1.5 hours after stimulation. We still see cognitive effects of ultrasound. We use K-plan in order to predict where to put the transducer, in order to predict how much pressure we get at the target. We have two different targets, inferior frontal cortex and thalamus. And we use a new fast pro system that you saw in an earlier slide, 500 kilohertz frequency, 20 milliseconds pulse duration, 200 milliseconds pulse repetition interval, a 10% duty cycle, 80% 80-second duration. And 10% duty cycle means that if we are effectively only stimulating for 8 seconds. Okay. So all we do is effectively stimulating for 8 seconds, but we do see a cognitive change 1.5 hours later. So this is the functional connectivity change that we see, the kind of biological change that we see in the system. On the left side, stimulating the inferior frontal cortex. On the right side, stimulating the thalamus. A blue color tells you that functional connectivity goes down, and a red color means that functional connectivity goes up for that particular connection. On the left-hand side, that for all other regions outside the target that where we see changes, we see reduced functional connectivity. And this happens at different time points. So for some regions, it happens early on, 20 minutes after ultrasound stimulation, whereas for other regions, it's only 30 or 40 minutes after ultrasound stimulation. On the right-hand side, if we look at the thalamus, we see a reduction for some regions, but we also see an increase in connectivity for other regions. The thalamus region at the bottom here is basically the contralateral thalamus, contralateral to the side that we're stimulating. So we see biological changes in terms of functional connectivity. We also see cognitive changes in terms of the performance in the stop signal task. So we can look at the accuracy. The accuracy is not changing regardless of where we are stimulating, but the reaction time is changing. So what in the middle is the reaction time before ultrasound stimulation. And then to the left and to the right, we have the reaction time after ultrasound stimulation. If you look at the right-hand part first, the yellow dots, this is stimulating the thalamus, and we don't see a change in reaction time at all. But on the left-hand side, if we stimulate the inferior frontal cortex, we see an 8% reduction in reaction time. So people get faster in reacting to the stimulus and correctly reacting to the stimulus. So the accuracy stays the same. So this is very interesting because you might think that people are already at the limit. You basically have to push a button. So you have the motor circuits. Those circuits take a long time. You have the signal going all the way from your brain to your finger. You have the whole visual system processing. So you might think that there is no big room for improvement. If you think about other studies that use brain stimulation, they usually see improvements of maybe 3%, maybe 5%. So having 8% faster reaction time is a big effect of ultrasound stimulation. If we then look at whether we can predict this change in reaction time on the x-axis here, we can predict this based on the change in biological features, so the change in functional connectivity between the target inferior frontal cortex and postcentral gyrus. So this connection is indicative. Okay, the higher the reduction in functional connectivity for that particular connection, the higher is the reduction in reaction time that we observe in participants. And we can also predict the reaction time change by the physical change we see in the system. So the physical change is the change in pressure at the target. So we can predict that based on the K-plan simulation. And the higher the pressure change at the target, so that the upper left side, the higher is the change in reaction time, the higher is the reduction in reaction time. So we did some mediation analysis. So we can predict the reaction time change based on the peak pressure or based on the peak pressure or mediated with the biological change in functional connectivity. We can also use computational modeling To give us some information about what is going on, we developed a MATLAB package or Octave package that is simulating brain tissue. So it could be any brain tissue, rodents, monkeys, humans. So we can specify what cortical layers are there, what neuron types are there, what is the morphology of neurons. And we then basically predict the local field potential. So what would happen if we stick an electrode into the tissue, or what would happen if you record fMRI from the tissue. So we can set up all those parameters concerning individual cortical layers or different neuron types and connectivity between neurons as well. We're using a simplified representation of neurons. On the left-hand side, you see the real morphology of the neuron, and then we use a simplified model with a lower amount of compartments, so maybe only 5 to 10 compartments instead of thousands of compartments. But even with this reduced model, we're able to reproduce what we see in experimental recordings. So what at the top is a recording of removed monkey tissue, temporal lobe tissue in rhesus monkeys. At the top of the image, you see the upper cortex, layers 2, 3, and at the bottom, you see the lower layers 4, 5, and 6. The color tells you the power in the gamma range, 20 to 40 hertz, higher gamma activity in the upper layers 2, 3. In the middle 4 time series, so those are recordings from those 4 recording points. So we have multi-electrode array recordings here. And on the right-hand side, you see a phase diagram. So you see this phase reversal between the top electrodes and the 3 electrodes that are below. And we see exactly the same pattern in our computer model. And again, the computer model was not trained to reproduce this. The computer model was only taking into account what is the anatomical information about the system, so what neuron types are there, what is the layer thickness, how are different types of neurons connected. And just based on this basic model, we see higher gamma activity in the upper layers, and we see the phase reversal between the top electrode and the lower electrodes. So this model can basically reproduce what we would see in experimental recordings of the local field potential. We then use this model to simulate what is happening after invasive stimulation. So this is sticking an electrode into the tissue and then using theta burst stimulation. And at the bottom, the electrode tip here, the electrode is going diagonally into the tissue. And you can see that there were changes in other neurons nearby. So we can observe changes in synaptic weight for other neurons here. And we can then also see which particular connections show changes. So connections between specific neuron types are showing changes as a result of this theta burst stimulation. We used the same model to look at non-invasive electrical stimulation. So this is simulating TDCS. So in this case, again, we have a cortical tissue. At the top, you have layers 2, 3, and then at the bottom, you have layers 4, 5, 6. And we can then have TDCS, the electrical field, going through the tissue in different directions. So from the bottom to the top, 0 degrees, or the opposite direction, or just going laterally across the tissue rather than going through the tissue here. And we can then look in the computer model what happens. So what change in membrane potential would we actually expect? So that preprint is online here. So we have the change in membrane potential, the DC shift as a result of TDCS stimulation. We have the intensity in milliampere of TDCS stimulation, and we have the angle, how the field goes through our target tissue. And what you can see here is if you have 180 degrees and 2 milliampere intensity, you get a certain DC shift as a result of stimulation. But then if you go to 135 degrees, so you go through the tissue diagonally, you need twice as much intensity, 4 milliampere, to have the same DC shift that you had before. So this might explain why brain stimulation is working for some patients and not for other patients, because the folding pattern of the brain is highly individual. So depending on the brain region, for some brain region, it's more similar between participants, but for other brain regions, there might be a gyrus for one person and a sulcus for another person. So you might have 180-degree direction of your TDCS field in one participant, but only 135-degree direction in another participant. And because of that, if you have 2 milliampere stimulation, it might work in one participant, but not in the other one. We also developed a computer model for optogenetic stimulations. If you have optrodes implanted and you just have a simple model with excitatory neurons and inhibitory neurons, you can basically simulate what happens when you have different stimulation parameters. So in this case, we have those triangular shaped stimulation pulses with LEDs. Or on the other hand, we have rectangular ones that are uniform and you have higher amplitude. And you get a different response of the tissue. So this was done in mice. And we get the same response experimentally that we get in our computer model. So we can use the computer model to basically predict what is going on. So just want to briefly talk about commercialization of new tech solutions and what are challenges in the field, in my opinion. So I think the current market gap is that the ultrasound device that we are using is not something you can take home. So you could potentially carry the new FAST system in a backpack and take that home, but you also need a new navigation system, which is less portable. So the new navigation system makes sure that your transducer is at exactly the right position on your head in order to hit the target. So both systems are first very expensive, so altogether more than $200,000, and you can't take them home. So it would be really useful to have some device that you can take home. And for this, we are working with New Gear. So I'm on the scientific advisory board of that company. And this company has a system called ZenBud that can stimulate the vagus nerve behind the ear, the auricular branch of the vagus nerve. And because it's just going for the nerve behind the ear, it's not going for the skull. So you don't need the same amount of power and the same equipment. And you don't need the targeting system. So you have this 1 centimeter wide an ultrasound disc in blue, where the ultrasound just gets emitted and it's just going in a straight line. And then you have a high chance to hit the vagus nerve. So this is really a system that you can use at home. Also have it over here. Okay, so I can maybe show it later in more detail, but it's basically a mobile system that could be used. But one market gap is to develop a system that can go for the skull, that is mobile, but that it also makes sure that you reach the deep brain target. So if you don't have a deep brain target, it doesn't really matter whether you're accurate. So if the target is very close to the skull, if your angle is slightly wrong, then you're still hitting the target. But if you have a deep brain target that is far away from the skull, then a small error in your angle will mean a big error deep in the brain, and it will mean that you're missing your target. So one obstacle for the future is to have some device that can accurately measure whether you're on target or not for deep brain stimulation. Coming back to the portable non-invasive vagus nerve system, the company did a pilot trial in anxiety patients, so with an anxiety index score above 16, and 85% of participants were responding to the intervention, which is more than you would get from other vagus nerve interventions where you just have 65% or so responding. And what we see with the system is a rapid reduction in anxiety scores in patients. So participants are using the device at home for 5 minutes a day, and they can choose when they use it. So it could be any time, could be in the morning or could be in the evening, and just 5 minutes a day. And then after 1 week, and after 2 weeks, 3 weeks, and 4 weeks, we basically measure their anxiety scores and also PTSD scores, depression scores, and sleep scores. So for those anxiety patients, you see this big reduction of almost 50% just after 1 week, and then anxiety goes down even further. And this image is including participants whether it doesn't work. So it does include the 15% non-responders. If we would exclude the non-responders, the results would be even more impressive. So for some participants, anxiety goes down to 0 after using this device. So you can see on average, anxiety goes down from severe to mild. For PTSD, it goes from moderate to mild, and moderate to mild for depression as well. For sleep, there's no real change, so people already slept quite well beforehand, in other words. So it might be different for patients that have sleep disorders, that there might be an effect there. But for anxiety patients, we do see this big reduction in anxiety. So I know lots of you are fans of invasive technology and implants. So the question is, why not invasive technology? So why not develop implants like Neuralink, for example? And I just briefly want to summarize this, but I'll go into more detail. So the selling point of implants is basically that, okay, you have a relative, maybe an adolescent with a mental health condition or young adult, and there's some kind of condition, so let's just put a brain implant into that kid. And the problem is that the surgery risk is 4.5%. So it varies on the procedure. It's like between 2% and 20% of brain surgery patients have an infection and those side effects because of the surgery. 4.5% is the level you would get for normal cancer surgery if you have a brain tumor and you just remove the brain tumor. So it's basically at the lower end of what you would have for an infection. And if you have a device or electrodes or batteries that have to be replaced, let's say every 10 years, it means that you need surgery multiple times. So if you have a Parkinson patient that dies within 10 to 20 years, it's not something to worry about maybe. But if you have a young person who is still living 50 to 60 years and you have to have another surgery every 10 years, you do get a higher risk that there will be at least one bad surgery. So if you have 5 surgeries, there was a 20% chance that at least one of those surgeries goes bad and you do have a brain infection that you have to deal with. So in terms of implants, in many cases, the outcome is that is not going to happen. So this was a general overview about invasive technology, but let me just go into detail. So surgery has potential side effects. So ethical approval is only possible, first of all, in adult patients. You would never do that in children. But then if you think about using that for healthy participants, like in a Matrix movie, everyone has an implant, it's highly unlikely that this will get approval. According to Christoph Koch from the Allen Brain Institute, he said it's very unlikely that the FDA will ever approve implants in healthy individuals. Let's think about the total addressable market for invasive neurotech. So let's say maybe 6% of patients would get implants at one point. And this means that 2% of the total population would have implants if we say that 1 out of 3 suffer from brain or mental health conditions. So 6% of patients or 2% of the total population, that's the market for brain implants. But of course, this market will decrease in the future as soon as other techniques become available. Other techniques could be digital tools to train the brain. It could be some cognitive therapies that become available, or it could be non-invasive technology. So currently, the addressed market share of implants is 0.01% of patients. So 1 out of 10,000 patients suffering from a brain or mental health condition has an implant. So this is including deep brain stimulation implants. It's also including cochlear implants. If we look at the most successful area for implants, Parkinson's disease, 1.4% of all Parkinson's patients have a brain implant. On the other hand, the market share will increase, even in the best case scenario, will increase very slowly, maybe by 8% a year, not due to the technology, but due to lack of neurosurgeons and centers with expertise in using brain implants. So it might be a bit easier to use robots for surgery, but still you do need centers that have neurosurgeons and that have experts in implant technology. If we historically look at deep brain stimulation over the last 15 to 20 years or so, you do see an increase in the number of procedures, an 8% increase per year. This is a linear growth, not an exponential growth. Okay, so it's a linear curve, which means that it will take a lot of time to go from 0.01% of patients to 6% of patients. Okay, so it's a very slow process, and I don't think we can expect implants to have a big impact in the field for brain and mental health in the future. So in summary, ultrasound neuromodulation is preferred by patients and the public. Biological ultrasound effects are indicative of cognitive effects that persist for at least an hour, showing potential for cognitive improvement, faster reaction time while retaining the same accuracy. Cognitive improvement can also be predicted from the physical effect of ultrasound, which is the change in peak pressure at the stimulation target. And this is a first step towards predicting dynamic effects, so both immediate effects during the stimulation and also long-term effects, hours or days or weeks or months after the stimulation. We're basically working on some computational prediction models to predict what is actually happening in individual participants. And commercial activity is really crucial to realize the potential healthcare benefits of new technology solutions. There are no big companies at the moment in the field, so it's really a good starting point for new companies. And it's really important to have reliable companies in the future that can support the field. There was more information in the book here, Changing Connectomes by MIT Press. This book is looking at how the brain is organized as a network, what are the main features of the network, how are network features linked to cognition, how is the network developing, both for healthy development, but also for neurodevelopmental and neurodegenerative. And it's also looking at the effect of learning and brain stimulation in terms of network changes. And I would like to thank my team, current postdocs, PhD students, former members of the lab who moved on to faculty positions, collaborators, the funders. You can follow me on X and Blue Sky. And I would also like to highlight that we currently have 2 open PhD positions and several postdoc positions. So if anyone is interested or knows of candidates, please get in touch. So thanks for your attention.
Thank you so much. That was fantastic. I really appreciate it. That was a lot to digest. And I'm so glad that we got that other perspective as well, especially on the non-invasive parts. We've had a few folks on before that worked on ultrasound BPIs, or ultrasound neuromodulation. And I think it's really just exciting to see how much is happening in the field and how big the effect sizes are. It's really quite incredible. I can't believe that not more people are getting more excited about it, like across larger population sizes or something, because the effects really seem quite large with like pretty small downside. So thanks a lot. That was really useful. Okay, if you guys have questions, feel free to raise your hand. I know that we have one in the question. Nicola, you asked your question in the chat. If you want to unmute, feel free to do so to ask it. Otherwise, I can ask it if you're not a good spot.
Sure, yeah. Thank you for the talk. It was really excellent presentation, Marcus, as always. I loved your comparison with the invasive technologies. I fully agree with you. And that's why I have a question, how you would compare the ultrasound stimulation with TMS, another non-invasive technology. And what I'm interested mostly is the precision and the portability of the devices. I think that's where the main differences might be.
Yeah. So in terms of comparison, so my own experience is not TMS, but I work with a colleague, Jeong Jung in Nottingham, who is a TMS expert. And she recently moved into ultrasound. So she's looking at the motor system, and according to her, ultrasound gives better results and more consistent results than TMS. So I think that's one benefit, that you can go for deep brain structures, but you can be very consistent in terms of how much energy goes to your target. Another part of the comparison is to really look at safety. So if you look at TMS, you often have side effects. So depending on where you stimulate, of course, worst case scenario, if you go to the cerebellum or over the neck and so on. But just overall, if you look at adverse side effects for TMS, 30% of participants on average have some kind of dizziness or headache or some kind of bad feeling after TMS. For ultrasound, that is 3%. And those 3% have very mild adverse effects. So it's something like feeling a bit strange for a few minutes. There were fewer side effects, and the side effects are less severe compared to magnetic stimulation. And because of that, ultrasound is a lot more useful if you go to children and adolescents. So it's much easier to get ethics approval in children in the future because of the safety and because of the reduced risk of side effects.
Could I piggyback on the question in terms of cost and portability? How do they compare there?
Yeah. So the TMS system is a bit less portable, but the main thing is for both systems, you need a new navigation device. So that's highly unportable. And then for the TMS system, you have something that is maybe 3 to 4 times the size of the ultrasound system. But the main challenge for portability is the new navigation system that is needed.
And cost-wise?
So cost-wise, at the moment, the ultrasound system is a bit more expensive. So the new FAS system that we are using, which is the main system that the labs are using worldwide, I think at the moment there are maybe 200 systems that are used. So some labs have 2 systems, so you can stimulate 2 targets at the same time. So there are 200 systems on the market at the moment. There were 3 big companies that are offering those ultrasound systems. I guess it will become a bit cheaper in the future. So I think if more labs are using it, the price will go down. There is also some hope that there was some engineering benefits that will make it cheaper in the future. But at the moment, it is more expensive than TMS.
I don't know if Alison disappeared from the connection, but I'll ask my second question. It seems to me that also with TMS, there's a limitation in the size of the coil that you probably cannot reduce it to as little as you can have with the sound sources. But do you need calibration in ultrasound? So in TMS, one of the problems is you have to calibrate to the responsiveness of the brain. You have all those motor things to do the calibration. Do you have to do the same with ultrasound?
We don't have to do that because we basically use the K-plan system to simulate how much energy we need and where we need to go in. So we don't need this calibration step.
Well, I think we also had Jake with a question.
Yes, hello. Thank you for that talk. It was really interesting. As always, I've seen you give talks before and they're always fantastic. I wrote lots and lots of questions, but I'm going to try and maybe choose some of the best ones. So for the survey paper, I was looking at the supplementary materials and I was quite surprised to see that there didn't seem to be much of a difference in people's perspectives on different neuromodulation techniques with age. I was expecting younger people to maybe trust technology more or something like that. So how do you see people's perceptions changing over time, given there doesn't seem to be much of an effect of age?
Yes. I guess one question is, okay, so one part is that you do something to your body, right? Which could also be piercing or having an ID chip in your hand, so any kind of intervention. For piercing, that did go up over the last 50 years, but I think it's different when you go to the brain. So when you think about putting something into your brain, there was still hesitance. And despite everyone watching Matrix movies, I think there was still a barrier in order to do that. You can even see that for cochlear implants, right? Some people have hearing loss at a relatively young age, in their 40s instead of 50s, 60s. And still people are hesitant to use cochlear implants. So only 7% of patients with hearing loss have a cochlear implant. And in some cases, because they don't agree to have one. So I think I agree it's surprising. You might expect younger people are maybe more willing to try something novel, but I think this kind of hesitance might not change much with age.
Yeah. Okay, that's interesting. The other thing was for Vertex, do you plan to add ultrasound within there? I know the mechanisms are harder to simulate, so it's tougher, but
we plan to get funding for it. We don't have funding yet. We just found out yesterday that we have funding to use to develop closed loop ultrasound and closed loop TMS. So that will be interesting to see whether closed loop stimulation is even more effective than open loop stimulation in healthy participants and in patients. But yeah, we don't have funding to extend the model to ultrasound yet, but it would be on our wish list.
Okay, great. Thank you.
Yeah.
Jake, if you have another one in you, you're welcome to go for it. And then afterwards, we go to Steve Potter.
Okay. Oh, so of the many other questions, one of them was about multimodality. So do you have any perspectives on using ultrasound and maybe temporal interference to hit a deep brain target with two different approaches, electrical and ultrasound at once?
Yes. So actually, that's something that we're trying now for closed loop. So we use temporal interference to entrain deep brain structures, and then we use ultrasound or TMS to stimulate either in phase or out of phase. So we just got the funding for that. We'll start to do it from March next year.
Okay. I look forward to that, the
Yeah.
results of that. Thank you.
Yeah. Okay, thanks.
Very cool. Okay, Steve.
That's very exciting stuff. I'm wondering if you could tell us a little bit more about when you do closed loop, what is the parameter that you're measuring to control the closed loop?
Yes. So there are different ways how to do that. So one possibility for closed loop would be to measure ongoing brain activity, to look at the phase of ongoing brain activity, and then just to stimulate in phase or out of phase. That is not what we are doing. So we did closed loop optogenetic stimulation in monkeys before when I was back in Newcastle 3 years ago. But basically for humans, we choose to entrain ongoing oscillations in the brain with temporal interference. So we basically enhance ongoing oscillations, let's say 20 hertz or 40 hertz. And because we use temporal interference for entrainment, we know what the phase is because we are stimulating with that particular phase. And once we know what the phase is, we then use ultrasound or TMS either in phase or out of phase to temporal interference to see what the effect is. So that's our closed loop approach for humans.
Fantastic. Anyone else, please feel free to raise your hand or drop your question in the chat if you can't ask it yourself. Otherwise, I'm going to ask a few questions that most people at Fawcett are always interested in.
Okay.
You can stop me by raising your hand and asking your own question. But so one thing that always comes up is like wanting to know a little bit. I guess you've already talked about it to some extent, but what's on your horizon in the next like year or year and a half? If you had all the funding that you'd want, what would you want to achieve? And then specifically what people in this group can help you get along. So for example, sometimes people are fundraising, sometimes people are looking for collaborators on a specific issue, sometimes they're looking for a specific contact that could give them access to an enabling technology, anything like that.
Okay, so Alice, I think you're gone. So I'll just start. So in terms of what would be ideal in the next 1.5 years, so one part is seed capital and venture capital. So I mentioned the Zenbud system by New Gear, where we have pilot results that we can reduce anxiety in patients. And the challenge to get that into the clinic is really to go for the FDA process, to get regulatory approval, and then to license this as a medical device. And that is very costly. So it basically needs millions of dollars in order to go through the whole FDA, RCT, clinical approval stage. So it would be very helpful to have funding for that. So that would be one challenge. The other challenge is what I mentioned in my talk, to have a mobile system. So at the moment, our system is not mobile, but if you have something that patients can take home, so they don't have to come to the hospital, this means that we can treat a lot more patients because there's a limited number of clinicians and nurses that are around in the hospital. So if we have a mobile device, that will be a big game changer to have transcranial ultrasound that you can use at home anywhere. Oh, I can't hear you.
Sorry. Everything went well in the next 5 years. Where do you think we would be in the next 5 to 10 years?
Yes. So ideally, we get rid of those pharmaceutical drugs because they're really awful. You have some drugs that have some beneficial effects, but they increase the risk of suicide. You have drugs that lead to obesity. If you look at schizophrenia, antipsychotics, they do get rid of hallucinations, but then they also lead to cognitive decline, which means that people are unable to go back to work, the memory performance is not working well, and so on. So the long-term goal, at least for my lab, is really to get rid of pharmaceutical drugs and to replace them with brain stimulation. It's a big challenge, and you have all those big pharma companies that have lots of funding for pharmaceutical drugs, and we don't have near the same level for non-invasive brain stimulation. But I think that's really what patients want, and this is a huge opportunity to have better treatment and fewer side effects and people going back to work even if they have mental health or brain health conditions.
And you mentioned at the beginning, I hope you can hear me now. You mentioned at the beginning the IQ point, basically, that it can measure
Yes.
like with up to 3 points off possibly.
Yeah.
Are there any like things on the horizon where we can actually improve patients, like not only like normal mental health if they already have a negative condition, but we can actually make also people that have a pretty high baseline very happy, or we can improve their cognitive function in specific areas. Is there anything like that in the next 5 years where you
Yes,
see?
I think that's possible. So there are already papers out there by colleagues where you can improve mood in healthy participants. Okay, so it's not patients, but healthy participants. They get more jolly after a few minutes of ultrasound. So that's already there. And I think it's entirely possible to improve cognitive performance. So one thing that we want to focus on for schizophrenia is to improve working memory. But then if it works in schizophrenia patients, it might also work in healthy participants. It might also work in dementia patients. So there could be a larger market for improving cognition in people. So I think that's entirely possible in the future, but it does need a bit of development.
And are there specific regulatory hurdles or something that one would need to overcome for this? You said, for example, for the invasive devices, this would probably never be, or like that the Allen Institute person said that this might never be allowed for healthy patients. Is there something similar on the horizon for the non-invasive and specifically the ultrasound bits, or?
Yeah, so it is a bit easier, but unfortunately, at least in the European Union, there's a problem that non-invasive brain stimulation is now a class 2 device, so basically, even though it's like mostly harmless, it's now in the same bracket as electroshock therapy or something. So it is a bit disappointing. So because of that, a lot of devices on the market are not marketed as a medical device. They're marketed as a wellness device. So it's just generically you will feel better, even though we know there are specific improvements that might be useful for patients, but we can't say so in the marketing. But of course, the long-term goal is to get FDA medical approval and to really market this as a medical device and then use it in the healthcare sector. But the cost is really the main hurdle to really go through all the trials and to go through all the processes.
Thank you so much for your work on this, and I really hope that we'll get there. I think the momentum is definitely forming. I think it's definitely spreading the knowledge that, hey, there is this thing that could be incredibly beneficial for a fraction of the cost, a fraction of the invasiveness, with a good safety profile, etc. So thanks a lot for your work on this, and I hope that we get to speak to you again in the not too distant future to hear an update on your work. So thanks, it's really exciting. Thanks a lot, and thanks everyone for your great questions.